Alright, so now we're going to talk about enzymes. We're going to go over 5 specific types of enzymes, but just to make sure we're on the same page about what these are proteins that catalyze chemical reactions. So they catalyze chemical reactions. These are catalysts, but they are proteins that are built into catalysts. So you can have something like a metal like platinum, that I talked about in the last video, that catalyzes a reaction. That's not an enzyme, that's a metal catalyst. A protein catalyst, we just call it an enzyme. So there are 5 major types we're going to talk about in this class. And so I'm going to list them out. I'm going to give examples of the types of reactions. So generally, a different type of enzyme is going to catalyze a different type of reaction. So we've talked about a number of different reaction types in this class already. And there's going to be a few new ones I introduce, and there's one that we're going to talk about a lot more in the next section. So I'm going to very briefly talk about it now, and then we'll kind of circle back to this idea in the next section as well. So the first one, kinase. So we've finally gotten original with the name. So the name really doesn't make a lot of sense. It's not going to help you figure out what this does. So this is a type of enzyme. Remember, they're proteins, so all their names are going to end in ase. So all enzymes have names that end in ase. We should put a note there. So if you see something ends in ase, just like if it ends in ose, it's a carbohydrate. If it ends in ase, it's an enzyme. So it's kinase. And this catalyzes phosphorylation reactions. And I'll remind you what a phosphorylation is, but it's just a condensation with a phosphorus. If you don't remember what a condensation reaction is, I'm going to remind you. So here we go. So I'll give you the name, description, and an example. We're just going to do this 5 times. Bam, bam, bam. And then we'll sort of frame the strike zone at the end a little bit too, because it's a lot of information here. So if I have something that can perform a condensation reaction and then something else that is a phosphate, well, then I can get a condensation reaction between the two. And so we can have a condensation reaction here. This is the type of reaction that kinase would catalyze. It would typically be a larger molecule, something more like a sugar, but I don't want to draw all of that out. So just to remind you what type of reaction this is. And so we get something like this, and a water. So kinase will speed this up. That's what it does. So instead of just trying to get them to bump into each other the right way, kinase guides them together, boom, water pops off, large molecule forms. That's what happens. How do you remember kinase? You just have to memorize that one, unfortunately. The other ones, the names will be more helpful. Isomerase. Isomerase. Well, this is going to catalyze switching a molecule between isomers. Quite often a carbohydrate. So I will actually draw a full carbohydrate on this one. I won't be as lazy as kinase. But catalyzes changing the isomer of a molecule. So it just rearranges an individual molecule. So for example, we could have something like a carbohydrate. Maybe you saw this. So there's carbon, 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 CH2OH. I don't know if I did this representation before. I think I wrote the carbons out, but it turns out, surprise, there's an easier way. Okay, then our reaction happens. So our isomerase would catalyze this process. We've got a carbohydrate here. We're going to have a very similar carbohydrate at the end. But now I'm going to put all my OHs on the right. So it's a different isomer. It's a different molecule, but it is very, very similar. So all it does is switch that H and that OH, which actually involves breaking a bond and forming new bonds. So it is a chemical process. The molecule has been rearranged, but it is now a different isomer. So it's same number of carbons, same number of hydrogens, same number of oxygens, and they're almost put together in identical fashion. Just a small, you just basically flip the switch here. You have different molecules. So isomerase does that. And so that'll let you do things like convert galactose to glucose or something like that. Like very similar sugars can get switched around. And that's good because, we have mechanisms to use glucose in respiration, but not galactose directly. But if we turn it into glucose, then we can. Alright, or same with fructose, if we can turn that into something. So, next we have the best named one. I'm serious. This is oxidoreductase. And it sounds like the name is straight out of a Harry Potter book or something. Oxidoreductase. So this one's a little tricky to talk about right now because it involves oxidation and reduction reactions, and if you're thinking those don't sound familiar, they shouldn't, because they're in the next section. So they catalyze, and if we put oxidation and reduction together, we get a shorthand for it, because why not, right? What's the point of taking a chemistry class if there isn't a shorthand for something? And that shorthand is redox. So catalyze redox reactions. And there's going to be two types of redox reactions we talk about. Hydrogenation and dehydrogenation are one. And then just changing the number of carbon-oxygen bonds is another. So oxidizing means increasing the number of those bonds in a molecule. Reduction means reducing the number. And that's the 10-second description. You're going to get a whole video on it in the next section, so don't worry. But as an example for now, let's say we have something like this, and we turn it into something like this. And this isn't even balanced. Alright? This is an oxidation, because we've had one carbon-oxygen bond, now we have two. So the increased number of carbon-oxygen bonds, it's an oxidation. This is one example. There's, 4 varieties. 2 types of oxidation, I want you to know, 2 types of reduction. But we will talk about that more later. Alright, so that's number 3. Number 4, so number 4 and 5 have very similar names, which might trip you up, so just hang in there. They do very different things. So, in terms of chemistry. So, that's helpful. Very similar names, very different results. What could go wrong there? So hydratase is a special type of oxidoreductase. So it catalyzes oxidation of a double bond with water, so it makes an alcohol. So for example, I'm going to draw the simplest double bonded carbon molecule I can. There you go. I need two carbons to make a carbon-carbon double bond, and no more. So this isn't necessarily a biologically active reaction here, but just an example of the overall type. And so what I get is 3 hydrogens on one side. Now I have a single bond, and I have an alcohol over here. So I take some of the double bond and I turn it into an alcohol. That's what hydratase does. And that is really important biologically, that type of reaction. We'll talk about that more later. And then the last one, hydrolase. I wasn't kidding. The names are very similar. So you got hydratase and hydrolase. Now, luckily, this sounds really, really close to the reaction it catalyzes. There's a type of reaction that you learned that sounds a lot like hydrolysis. Okay? Hydrolysis. So this catalyzes hydrolysis reactions. And so, for example, we could have, something like, I always like to draw, I like this one. because it looks like a bat. You've seen it a number of times now. It's my go-to thing that you can hydrolyze. All right, and a water to break it apart. So remember, hydrolysis is going to take this and it's going to turn it into two molecules. So we got carbon-carbon, oxygen, but now that's an OH. And we've got an oxygen that's going to be an alcohol as well, carbon-carbon. So that is a hydrolysis reaction. And it's one you've seen before. So it's going to be catalyzed by a hydrolase. Alright. Now, I just dumped a lot of information on you, and some of it you don't even learn until the next section of the class, really. So, in terms of what are you supposed to do with this knowledge, the answer is just, if you see a reaction, what type of enzyme would be catalyzing it? So, think of it as kind of like a fill in the blank. So you can have your enzyme type here. I'm going to give you products and reactants, okay? So you don't have to fill that stuff in. And a lot of times when I give an in-class test, it has a format like this. Okay. And we've got our reactants. I'll write a fresh one below. And our products. So these are given. And then you just fill in the smiley robot face with a mustache or whatever. And so this is like if there's any other reactants, this is if there's any other products, and then this is the enzyme name right here. So let's put it to practice. So let's say we have something like, something like this. Okay. So you've got to fill in a name here. There might be something to put here, there might be something to put here, or there might be nothing at all. It might be blank. So we're starting with this, we're ending with this. That's what you're told, and then you might see boxes to fill in or whatever. What do we do with this information? Well, what type of reaction is this? Okay, what type of reaction do we have down here? It looks like we've added a phosphate to something. Okay, so it's a phosphorylation reaction. So that means my enzyme type, which I'm going to write down here, is kinase. So I'm holding off on drawing the boxes till I actually have the name, so I don't have to do anything weird like, oh, I drew a box too small with a Sharpie. Let me erase that and fix it, just so it's clear. So you would see something more like this format. Alright, so if I'm performing kinase reaction, is there another reactant here? Okay, am I reacting this with something? And if so, what is it? Well, phosphate. Okay. I'm just going to write the name. Okay, and so I'd have this plus phosphate gives me this, and is there anything else I get? Well, remember, this is actually a condensation reaction. So by putting these two molecules together, I also get water as a product. And so in this box you put any other products, in this box you put any other reactants, and we filled the whole thing in. We're good to go. So that's what we do. And if there is nothing, so for example, we'll do one more. Let's say I have, I really put myself in a bind here. CH112. Is that on screen? Yes. Alright. I have my reaction take place. You might already know where this is going. Obviously it's going to the right because I got to write my products, but you might already know what type of enzyme this is. Alright, so what have I done here? What type of reaction is this? Well, I just changed isomers, so it is an isomerase. And when I perform a reaction with isomerase, catalyzing it, are there any other reactants? No, because I'm just rearranging what I have. And so I would just leave this box blank. You don't have to write anything in there. And then over here, do I have any other products? Well, no, all my pieces are accounted for. There's nothing else popping off. I'm not like releasing a water or anything. And so I can just leave that box blank. And that's well answered, just like above. That's all we're looking to do, is just fill in the boxes as needed. You don't have to fill something in if there's nothing there. Alright, so that's the whole story on kind of what I expect you to do with this. We are going to spend quite a bit more time on redox reactions in the next section when we talk about reactions in general. And so I will try to revisit this format and bring oxidoreductase back into the discussion in the next section, because we also have to bring in the coenzymes like NAD plus and FAD. So biological systems have some pieces to help balance them out. So there was a reason I didn't show a balanced redox reaction, because I haven't told you the whole story to be able to do that yet. And so that's what we're going to work on next, among a few other things. So we're going to really kind of like lay down the groundwork of all the chemical reactions we've talked about so far. The number is growing, growing, growing. And then we're going to zero in on redox and talk about that one quite a bit as well. So you should be able, at the end of the next section, you should be able to tell the reaction types apart and be confident, you know, looking at products and reactants and saying what it is, and being able to put in these, you know, fill in the blanks on these types of things with a little more confidence. But for now, just understand these, what types of reactions go with these, and then if there are other reactants and products for the ones that aren't involving kinase, so, or not kinase, oxidoreductase so much. Kinase is pretty straightforward. You just have a phosphate in the water here. Alright, so I will leave it there. That's the end of this section. That should give you everything you need for the homework. And, well, actually the homework looks like it has FAD and NAD+, but that's not going to be talked about till the next section. So, oh, I'm looking at the homework for the next section. Durr. Alright. Yeah, so you'll be good. You can do the homework off of what I have. That really, that scared me for a second. Alright, so, yeah, next section we're going to build on this more. But for now, just have a basic understanding of the types. And keep this formatting in mind, because that's going to come into play a lot more in the next section. Alright.